Hello, my name is Likhit Tanishq and I'm from Little Miami High School, and my research question was RTM, Refined Tumor Masking for Precision Optimized Glioblastoma Multiforme Resection using augmented imaging. So, first of all, what is glioblastoma multiforme? Introduction. Glioblastoma multiforme is one of the most aggressive and lethal forms of brain cancer, characterized by its rapid invasive growth and irregular shaped boundaries. This highly malignant tumor can spread into healthy brain tissue, making it difficult to treat and surgically remove. As a result, surgical resection is challenging, leading to high recurrence rates. In response to these challenges, this project introduces RTM, Refined Tumor Masking, an advanced system that uses augmented imaging to enhance tumor visualization and surgical resection. By utilizing deep learning techniques and neural networks, RTM uses MRI scans to create highly precise tumor masks, and these masks assist neurosurgeons. Problems in GBM treatment. Unclear tumor boundaries. Surgeons try to remove as much of the tumor as possible to relieve symptoms and prolong life, but the boundaries between healthy and cancerous tissue are unclear. This makes it difficult to avoid removing too much normal brain tissue. Another is incomplete resection. A study by Johansson et al. Found that 35% of patients had complete tumor removal, and many experienced tumor regrowth within 6 months after surgery. Residual tumor cells not visible during surgery often lead to reoccurrence. The BBB. Blood-brain barrier is a mass of cells which block chemotherapy drugs from reaching the tumor, while GBM weakens parts of BBM, much of it stays intact. It prevents about 98% of molecules from reaching the brain. Next, abnormal vasculature. The tumor abnormal blood vessels and low oxygen areas make treatment even harder. Newer methods like nanoparticles that cross the BBB and ultrasound to temporarily open it show promise, but they're still being tested. Hypothesis. My hypothesis was developing the RTM system and integrating it with advanced imaging techniques can improve the accuracy of glioblastoma multiforme GBM tumor resections. This approach helps surgeons achieve more complete tumor removal, reduces the risk of reoccurrence, and improves patients' survival outcomes compared to traditional surgical methods. Research datasets. For the RTM, we used two research datasets, the Lumiere dataset, which contained 2,500 manually segmented masks in T1, T2 FLAIR, and CTI, which helped in imaging analysis. This contains 91 GBM patients, providing a robust sample size for analysis, 638 study dates and trials, offering longitudinal data for tracking tumor progression. The next was the Junsheng dataset acquired through online collaboration with the university. It contained 3,064 T1 weighted contrast enhanced images from 233 patients, 3 types of brain tumors, meningioma, glioma, and pituitary. Split into 4 subsets, each contained 766 slices, and in the Lumiere dataset, each contained 32 slices. These were provided in a MATLAB format .mat, which you had to pre-process to .png. Both of these datasets are crucial for training and validating our primary deep learning model. Image pre-processing. So this flowchart is how the image went through pre-processing and augmentation. So first, the MRI image from both datasets were resized to a 256 into 256 resolution, while preserving spatial structures, for example, the coronal brain structure. For Lumiere's NIFT formatted image, the slice depth was 32 slices per scan, while the Junsheng dataset was 766 slices per scan. Intensity normalization. Z-score normalization was used to ensure pixel intensity values had zero mean and unit variance across the dataset. Image registration. Augmented image techniques such as image registration, fusion, and data augmentation were used to enhance the quality and clarity of medical images as shown in our hypothesis. Image fusion. What image fusion does is it combines data from all three of these steps from the MRI and CT scans, and it creates diverse training sets by applying all these types of augmented imaging techniques, which enables for more precise tumor localization. The RTM system. We implemented the RTM system, a deep learning system designed for precise tumor segmentation, classification, and survival prediction in GBM. Deep learning architecture. RTM consists of a tumor segmentation model, which is a 3D unit to accept 3D pictures as well. It uses skip connections to capture complex tumor shapes as shown in figure 2.2. This model is trained to precisely delineate the tumor boundaries in MRI scans, crucial for accurate surgical planning and treatment monitoring. Next, the tumor classification model, which was a ResNet-based CNN, as shown in this image, which takes an input image, goes through all the architecture blocks into a neural network, and it classifies it as benign or malignant based on if it's GBM or non-GBM. A survival prediction model. What the survival prediction model does is it analyzes both spatial and temporal tumor characteristics and patient details and the tumor size, and it predicts the patient survival rates based on tumor spatial features and temporal changes observed in the MRI scans. What the fusion model basically does is it takes all the data from these three and it fuses it into one big model, so the model is more accurate, a unified analysis, and a holistic view of the tumor itself. As you can see, these three pictures are code blocks. The first picture is the 3D unit. What it does is it has an attention mechanism to enhance skip connections, which is a great thing to build more neural networks as the data passes. Next, we have a forward and pass the data to delineate the tumor. As you can see here, this is a ResNet CNN classifier. What the ResNet CNN classifier looks like, it loans the pre-trained model, it freezes early layers for better feature extraction, and it replaces the layer with the custom layer over here for better localization, and it prints the output shape and the details. What you can see here is the survival prediction model. What it does is it takes the data from all this and it predicts the survival shape and the results over here. Visualization and optimization technique. GradCam, gradient weighted class activation mapping, was used to predict and show the tumor over here, as you can see in the sample image. It targets a layer and the layer is undergoing the transformations to create this image. We use hyperparameter tuning and Intel 1 API and GPU acceleration to speed and boost the training with a better learning rate. As you can see in the figure, the inference time boosted up and it also showed outlier features. So what we did is we wrapped the entire code in this Intel 1 API class wrapper, and it increased the speed and efficiency of the model. So we have a web-based implementation. So this is a website I designed. Users can upload an MRI scan in DICOM or NIFT formats through a simple drag and drop interface. The platform validates file formats before processing to prevent errors. Once a MRI is uploaded, the system performs pre-processing as shown in the before steps to normalize the scans and ensure compatibility with deep learning models. The uploaded scan is sent through the Refined Tumor Masking system in the loading screen, which is optimized using Intel 1 API over here into one API to boost the training and give the optimized results to provide fast and accurate results. A results dashboard. A comprehensive results dashboard including detailed visual and numerical insights including tumor segmentation masks overlaid as you can see here. This is the tumor and the outsides are the tumor border. 3D visualizations as you can see in this picture was a test. It is still on the website but is a test in progress. As you can see here, this is the RTM Refined Tumor Masking website. Users can upload a file, and get the comprehensive results, the axial, sagittal, and the coronal, the spatial features of the brain. A flowchart. So this flowchart is what the model uses to follow. Users start with an MRI input. Once a MRI is uploaded, it goes through, is the image format correct? If yes, then it goes through augmented imaging and resizing. After that, if the image quality is sufficient, if not, it goes back to first step. If yes, it goes next. It applies the RTM workflow using Intel optimization, and then it goes is if the tumor is detected. If the tumor is not detected, it is flagged as no tumor detected. But if yes, it extracts the shape, size, and the tumor border. Goes next and classify tumor types. It determines if it's malignant or benign or GBM or non-GBM. It goes through a high-risk tumor. If it is a high-risk tumor, priorities for surgical planning, and goes through the rest of these flowcharts. It generates a tumor mask, as you can see, the grad cam, and it generates a 3D reconstruction if applicable, and it also gives a risk assessment, and it gives a segmentation overlay, a report, and it ends the process. Results. So RTM scored a 98.2% accuracy on tumor classification, and it scored a 0.96 accuracy on the dice similarity coefficient. Measures over. So the dice similarity coefficient is where the predicted and actual tumor regions. It's a test where it's overthrown on them and to compare the accuracy, as you can see over here. Intersection over union, it confirms the segmentation process. As you can see over here, we had 98% accuracy over here and a 0.07% loss over 100 epochs. The RTM system had a 98.5 grading accuracy in the tumor, which is the classification model, and the RTM system was trained for 100 epochs with a learning rate of IE3, given an equal balance between convergence speed and stability. From the outset, the models demonstrated strong and consistent performance with no fluctuations in accuracy and always constant up or down. In terms of segmentation, the intersection IU and all the other scores over here demonstrated the predicted tumor regions closely matched the actual tumor boundaries. Conclusion. The RTM system significantly enhances glioblastoma multiforme detection, segmentation, and classification through advanced deep learning techniques. By providing improved tumor localization, RTM enables precise boundary detection, which is crucial for surgical planning and execution. Future research and extensive clinical trials are essential to validate the system's efficiency. Future testing should incorporate a diverse range of MRI scans, including variations in image quality, scanning protocols, and patient demographics to ensure the model's robustness and generalizability across. So right now we only support MRI, PET, and CT should be applicable for revolutionizing GBM treatment and enabling more effective surgical resection, improving patient outcomes, and extending survival rates. So these were my references I used throughout the project, which helped guide building this project and the slideshow and the final research paper. We have a lot of good reviews over here, and thank you. Any questions I'll be happy to answer on my Gmail over here, and that's it. Thank you.